A study published today by an international scientific journal shows how the work developed by researchers from Campinas in Sao Paulo can help understand the functioning of the brain. Abelio has a type of epilepsy resistant to medications used to control seizures. He undergoes treatment, but does not feel secure. I am very afraid because, like this, sometimes I get nervous, and if I go out a little nervous, I am already afraid of having a seizure on the street or somewhere. One of the difficulties in treating not only epilepsy, but other neurological diseases is the complexity of the brain. Without a shadow of a doubt, it is the most complex organ in the human body and is still a relatively unknown organ compared to others because it is difficult to access. Inside our brain, there is a network of cellular communication full of ramifications with specific connections. Furthermore, there are several tools and dynamics that regulate all functions of our body. For the first time, researchers were able to visualize part of these neuron circuits in activity and how they are affected by epilepsy. It is a major breakthrough in understanding better how our brain works. Until now, it was necessary to physically slice parts of the brain to analyze the cells. Scientists at the National Center for Energy and Materials Research in Campinas used a particle accelerator to perform X-ray microtomography capable of showing neurons with precision. From this, they obtained three-dimensional images of the organization of cells within the brain. If we really want to search for cures for diseases, first, we have to understand exactly how they work. The results were published in the journal Nature Science Reports. Although the technique was used to visualize the region of the brain that triggers epilepsy, it also opens new perspectives for understanding how the death of neurons occurs, which causes diseases like Parkinson's and Alzheimer's. By quantifying these cells dying throughout the process of neurodegeneration, I can track the progression of the disease, and I can also know exactly which region of the brain is affected by the death of these cells. And maybe in the future, how I can control this neuron death.